the C9orf72 gene is a more complicated gene, and genes are made up of exons, which are the bits that code for the proteins to be produced, and then introns, which are the linking bits of the DNA that connect the coding exons. And in C9, there is a sequence of bases, 4 Gs, 2 Cs, that normally you have up to 30 of those in that intron. But in C9 ALS, there can be hundreds or even several thousand of those repeats. So it's a hexanucleotide expansion in a linker sequence, an intron of the gene. And it took a long time to find that. And what happens is that expansion gets made into a messenger RNA, and normally or a pre-messenger RNA, normally that linker sequence should be cut out in the nucleus, and then just the coding parts of the C9 gene get out of the nucleus into the cytoplasm of the cell where the proteins are made. But that linker sequence is sticky, so it sticks to certain RNA binding proteins, and sticking to those proteins, instead of it being disposed of in the nucleus, that stickiness and sticking to particular nuclear export factors mean that abnormal sequence that should be disposed of in the nucleus is getting out into the cytoplasm and getting to the ribosomes, which are the factories where proteins are made. And that abnormal sequence forms these toxic dipeptide repeat proteins that are toxic to motor neurons. And the 2 antisense oligonucleotide trials that were supported by Biogen and then another company called Wave Therapeutics, that expanded region can be read in 2 directions. So it can be read in a sense direction going one way and an antisense direction going the other way. And because of that bidirectional reading, there are 5 different toxic dipeptide repeat proteins that can be produced. And the ASO in both trials only tackled the sense strand and leaving the antisense untackled with, if you like. And so we think that may be a major reason why the trials weren't effective. We don't know the full reasons, and the publication relating to the Biogen trial, I hope, will appear in an important journal very soon. We've just done the 3rd revision of it, so it should be out soon. But that's the major thinking, that it probably didn't work because the antisense strand wasn't tackled, and there's some evidence that the proteins produced from the antisense read are more toxic to motor neurons. So it's kind of back to the drawing board to try a new approach for that condition. Very important, because the C9 gene change causes not only ALS, but it can also cause frontotemporal dementia. So we haven't given up on that, but we haven't got the right therapy for it yet. And it's a more complicated gene in some ways than SOD1 is.